Here. Okay. So testing. Is that working? Yep. How about if I'm speaking like this? Yeah. Great. A little bit louder. Where's the volume button? Oh, that's down there. Okay. Well, there is something here. I wonder if this is just the volume. Oh no, that's something else. Is the volume okay now? Not sure if we found that button yet. Sure, I'll turn the volume. It's not on that remote control. No, something else. Okay. Let's see if this works. Okay, yeah, I'll move it up a bit. Here. Like this. Okay. I can also try to speak up. All right. Right, so let's get started.
One, two. So welcome everyone to to our CSC colloquium. So today we have Professor Randal Koene. So he's we're delighted to have him over to talk to us about whole brain emulation and how we can basically validate functional, like MRI functional measurements with the framework that he's proposing. So Dr. Koene is a neuroscientist working on the functional reconstruction of brain circuits via neuroprosthesis and neurointerfaces. He introduced the concept of whole brain emulation and is a lead architect of the scientific roadmap for technological development. He's the founder of Carbon Copies Foundation and was a software architect of high-throughput electron microscopy company Voxa, which provided the electron microscope technology that acquired the 10 petabyte cubic millimeter mouse brain connectome data recently published in Nature. He leads the development of a platform for brain emulation challenge to accelerate advances towards building functional models based on collected brain data. Dr. Koene's publications are available, and with this, we'll get started.
Thank you. Thanks very much. And thank you for inviting me here. I'm honored to be able to speak to you. All right. So the title of my talk is Reverse Engineering the Brain, Leveraging Fully Known Ground Truth to Accelerate Connectome Functionalization. And there was an abstract we sent out, which I'm not going to read it. You probably received it by email, but you'll see what's in the talk anyway, so we don't have to read through this. There are 6 main parts in this talk. First, I'm just going to get the who am I, what am I associated with out of the way. Then I'll talk about whole brain emulation a little bit to explain what is meant by that. Then talk about connectomics and how connectomics gives us an opportunity to really get into what some of us are calling causal neuroscience as opposed to correlational neuroscience. Then a bit on functionalizing connectomes, attempts that have been made there, how you use synthetic data to try to improve ways to functionalize connectomes, and then a bit about our brain emulation challenge platform, the software that we're developing, try to help everyone do these functionalizations. So just a brief bit about who we are. Carboncopies.org. This is the front page of our website. This is a 501c3 nonprofit research and education organization. And the main aim is that we are interested in this concept of whole brain emulation, and we're trying to advance research in that direction, trying to advance understanding of what that could be. On the education side, so what we're doing, as I said, is we're trying to raise awareness about the cutting edge research and development that's been done on brain and mind that heads in that direction. We also try to educate, depending on the level of the say the group that we're talking to about with whomever I'm describing it to, trying to really get an understanding going about how fundamental all of that is to us. So how fundamental mind and brain really are, in the sense that they are the world that we live in, that everything that we experience every day is only that which our brain can process and so forth, and how important it is for civilization and exploration and everything from here on out. I would put fully understanding the brain and being able to work with it, just like you can work with any device that you understand completely, to be something of a deep frontier along the same lines, as important, as difficult, as interesting as, say, really the fundamentals of physics or cosmology. And so we try to also educate about the possibilities and significance of a complete understanding like that and reconstructing the brain, and to help explain this new field of brain emulation. We ourselves, we believe that whole brain emulation is something, is a technology, a development that is essential for the long term of a civilization that's really intent on bettering itself. On the research side, we have three main projects going right now. One is an ongoing project that's been going on for 10 years and more, because we keep on updating the roadmap to whole brain emulation. The second project is an ethics framework. This is a project we started maybe 6 months to a year ago. And in that ethics framework, we're trying to kind of get ahead of the curve a little bit and to explain to researchers their responsibilities and what they should be aware of. We develop technologies in the direction of whole brain emulation. Also, what the situation is for potential patients who might undergo procedures where emulation is involved, and what it means to a society, technology like that. And then on the software side, we've got this Brain Emulation Challenge platform. That is a platform that is developed to create synthetic data, artificial data sets that aim to be, in many ways, very similar to the real data sets that you obtain through recording, microscopy in the brain. And the intent of those is to accelerate methods of development towards being able to convert data you collect from the brain to working models, making this really big step happen. If you want to see more about that, you can go to braingenics.org, which is the website where we're keeping all of that in place. So we're an open source organization. Everything we do is available to everyone under a public license, and we're driven by citizen scientist volunteers. They have all sorts of backgrounds, and we're always looking for more. Computational neuroscientists, of course, programmers, people who are interested in or have experience in machine learning and AI, philosophers, mainly philosophy of mind, writers, web developers, event organizers. We have opportunities for PhD dissertations at the organization. We've had some people who, together with us in collaboration with the university, carried out a dissertation while working with carbon copies on the topic of whole brain emulation. There are always internships available, and there are students working with us at every level. As you can see, we're also pretty international. Most of us are located somewhere in North America, but we also have a fair contingent in Europe, people in Asia, Australia, and in Chile. We work together with many others, of course. I'm listing a few of the collaborations that either have been there in the past and that we still have connections with, or people working with us right now, who also provide us with expert advisors. There is Genelia Research that you might have heard of. They're a brain research organization on the East Coast. The Allen Brain Institute up in Washington. Max Planck Institute, Jülich in Germany. They have locations in many places, but it's specifically this one that's working with us. eBrains, which is the computational side of the Human Brain Project. It's where they put all their big models. There's a lab at the MIT Media Lab that is working with us. Of course, UCSC and UC Berkeley, USC. There are two labs there, Ted Berger's and Dong Song's, who are working with us because they work on a prosthesis for the hippocampus. And there we also have affiliations at the medical center. There's a fantastic lab, University of Pennsylvania, that you'll hear more about in this talk. University of Oxford, we have an advisor from there. University of Melbourne, Nottingham, the Horizon Center, they worked with us on those PhD dissertation possibilities. And then there are some non-university organizations like the Brain Preservation Foundation, which is run by a friend of mine, Ken Hayworth, who works at Genelia, Foresight Institute, and then companies like Eon Systems. So if you want to know more about that, if you want to get in contact with us, if you want to learn all sorts of things that are not going to appear here, please just check us out on our contacts, or maybe you can just Google us, Google Whole Brain Emulation or Google Carbon Copies Foundation. You'll find a whole ton of things. All right, so whole brain emulation. There's a kind of a philosophical foundation to it all, or a foundation that has a few assumptions that come along with everything we're doing. And for one thing, we try to understand what the brain is and what it isn't. In other words, we try to understand its limitations, but also its purpose. And for that purpose, if you look at it, one thing we can say is, well, it was evolved to coordinate the body. You can also say it's evolved to model our world and to model ourselves. So what we're perceiving is a model that the brain is generating that gives us the world and gives us ourselves. And we try to identify where we are in that world, perhaps by noticing which things we can control. So it's not really about all of reality. The brain never shows us all of reality. It shows us that which it can process, that which it receives information about. It's purposeful and it's also very powerful. Probably still one of the most powerful computing systems that we know of. Then we make a distinction between simulation and emulation. So this is, I'm telling you this just so that you understand why we use the term whole brain emulation. Simulations are mathematical abstractions. It's when you're trying to explain something, you're trying to describe a thing in the world, and you use an approximation that you think is appropriate, what you're trying to describe. And emulation is a little different. It's not just a mathematical abstraction. It is something that is very purpose specific, and it's usually a re-implementation of some sort. Emulations often contain many simulations, but simulations don't necessarily contain emulation. Here's an example, for instance. Let's take the Nintendo Game Boy, emulating it on a phone. So what is the purpose that you have there? The purpose is you want to be able to play games. And you should be able to play the games in the same manner as you were doing before. It should give you the same experience. So the purpose is really to have a certain kind of experience. And you don't care how the original Game Boy was creating electromagnetic radiation, where it was warming up, those sorts of things. Those are not part of the emulation. So when you do an emulation, you could do it at all kinds of levels, but you pick the level that's appropriate based on the purpose. Let's see, working? If you're going to do whole brain emulation, there are some high-level things we should talk about. We already said a few things about philosophical grounding. The philosophy that is closely related to it would be functionalism. So the notion that what we are, what we experience, cognition, consciousness, all of these things are emergent properties of mechanisms. It's a very physical, materialist philosophy. And you have to know what your goals are. Describing the goals, oh, what just happened? Oh, that's not good. Oh, we're back. All right, that was interesting. Describing your goals means you have to understand what your success criteria are. When do you call something a successful emulation? Having success criteria is great, because once you have success criteria, then you can devise metrics, know how you can test if those criteria have been met. Constraints are super useful. We all know that there are constraints in all these systems because some things just get drowned out by noise. And for example, if you have populations of neurons doing something, then the activity of a single neuron might not matter very much. There are all kinds of constraints. Or in a computer, you know that one of the constraints is that things happen on clock cycles. Take that. Those help because they really reduce how many possibilities you have to explore when you're trying to describe this. Another really important thing about this description is scale separation. When you have a highly complex system, it's something that comes from complexity theory, the idea of scale separation. It's great if there is some way to say, for these purposes, for this thing that I'm trying to describe, for my purpose, I only need to describe the system down to a certain level. Below that, you could replace everything. You could put a computer there instead of biology, in our case. That's what we're looking for, right? So the question for a brain, for instance, is, do you need to consider molecular things? Do you need to consider atomic processes? Do you need to consider quantum mechanics? There are people who have quantum theories about the mind. Or is it good enough to model neurons and how they talk to each other? Then there's data acquisition. This is super important. High throughput, because it's an enormous amount of data, really. It's an enormous amount of material you have to churn through. You want to get down to the resolution where your scale separation works out for your purpose that you have. So you need to scan brains at a very high resolution, spatially or temporally, if you're doing recordings in vivo. And it needs to be done fast enough that you're not going to be recording for hundreds of years, which is easily possible if, say, you're working with a chimpanzee brain or a human brain or something like that. And there's post-processing of the data. Those are what we might call simple things like segmentation, denoising, and so forth. But then there's the really hard part. So this used to be the hard part, data acquisition, not as hard anymore as it used to be. I would call it an engineering problem now, not a scientific one. This one here, though, is pretty new. Trying to take the data that you collected here and building the model that works. Functionalizing. It's also called the translation problem because what you're doing is you have a pile of data. It could be voltages that you've recorded in a cell, or it could be the shade of gray of a pixel in an electron microscopy image. But the shade of gray is not what you put into your model. Your model is going to have response functions for neurons. Those are going to be exponential functions. You need to know what you're going to put in that exponent. You need to know your parameters. So finding the parameters is the translation problem. And then, of course, you have to be able to verify, validate, and then there's implementation. I've got a little list here of the animals that are used typically or that are thought as possible good targets as model organisms, because of course you don't want to start with the human brain. It's much too big. There's too much there, too complex. Right at the bottom, we've got C. elegans, the tiny little worm that only has 302 neurons and that has what is pretty much a fixed connectome. The neurons are always connected in the same way. It's not a great organism if you're trying to learn about human brains because the cells work differently. Those cells don't spike. They always work sub-threshold. So they're communicating in an analog way. Also, because there are only 302 of them, every one of those cells has a very specific purpose. It's almost like a brain region. So you need to know every detail about that neuron, get it just right. Next up, fruit fly. This one is the one you're going to hear the most about in this talk. That is a fantastic little organism that is just about the right size that we can handle it right now with electron microscopy. The honeybee is a good target as well, but not as well known, not as well understood. Zebrafish larvae are great because they're transparent. That means you can use techniques like calcium imaging or voltage imaging, where you can see through the specimen and watch neurons activate as the zebrafish doing things while it's alive. Then here's the mammal, the set of mammals. We've got the Etruscan shrew, which is the smallest mammal. Mouse is very well studied. And the reason is because we have an enormous number of tools with the mouse. We have genetic gene lines that are specifically developed so that you can make certain cells light up when they activate, they luminesce. And there are all kinds of other wonderful tools with the mouse. Rat, of course. A lot of scientists are now interested in using marmoset. Just fun, marmoset. They're cute, aren't they? And then there's the macaque. Of course, macaque is often used in neuroscience, chimpanzee, and then human. And you can see how those neuron numbers start to add up until you get to 86 billion. Take a little sip of coffee. Okay, very quickly, things you would do if you're going to do a whole brain emulation. First thing you do is you collect your validation data. Probably do that first because that data is probably data that you want to get from the living person or animal. It's something that you have to do beforehand, especially if the technique you're using for the big data collection is something you do post-mortem. Then there's sample preparation. And depending on whether you're carrying out the remaining process right away, or if you're going to be storing the sample for a long time, it's a different type of sample preparation. Then there's the acquisition of the data, the high throughput data collection. And here I'm talking mostly about connectome data. So I'm talking about electron microscopy on samples that have been prepared for high throughput electron microscopy. Then you decide how you're going to go about your modeling. You have to pick the right type of architecture. And this is where constraints already become very important. So you understand what types of activity matter and which ones don't, which types of noise do you have to care about, which ones don't you, and so forth. So you pick something. For example, you could pick a compartmental model. That's where you describe a neuron that has a couple of dendrites coming out here as compartments, each of which is basically a little electric circuit. And then you measure to see how thick is that dendrite to determine what is the capacitance, what is the resistance of that circuit, something like that. You might be building a network model, and there are many other things you have to select. After you've done that, this is the point where you start doing this thing called system identification, where you're trying to describe your system, the model that you've chosen to build, using the data you've collected. So this is where functionalization and translation comes in. I already just mentioned that, but you have to get into it. But for example, what you're modeling might be described by something like this. There's equations here for your circuit. And then somehow, from measurements that you're making here, or other bits of information, you have to fill that in. You have to figure out what those parameters are supposed to be. And then ultimately, verification, validation. And there you go. All right, so connectomics, causal neuroscience. So let me say something about most of neuroscience right now and up to now. I'm calling it correlational, and so do the scientists like Konrad Korting, I'll talk about him a bit in a second, who are looking beyond what most of neuroscience has done so far and trying to get to this causal neuroscience. Neuroscience is mostly correlational in the sense that the studies are set up to test correlations. And MRI studies are a very frequent example of that. And we could get into all sorts of critiques that are out there about papers that currently exist in MRI. I'm not going to do that right now. So a typical process in neuroscience is something like this. You start off with some basis of knowledge. You've got prior literature, or you've seen something before in an experiment, and because of that, you have some idea, hypothesis. You're thinking, you know what? I think the cells in area X, or these cells over here, are involved in this behavior Y some way. Or maybe your hypothesis is more complicated. Maybe it's something like cells in X talk to cells in Y, talk to cells in Z, and that causes behavior A, or whatever. So you set up an experiment where you're going to carry out some type of manipulation on this wonderful creature here. And then you record. And then after you've recorded and you look at that data, you discard or modify or keep your hypothesis. Right? So now you've made this correlation between X and Y, or you've discarded that correlation, or you have something to say about it. But let's look at the problem of whole brain emulation and even simpler things like just understanding what a particular circuit in the brain does, or how you get to, say, perception or something more elusive in cognition, like, say, consciousness. Could a neuroscientist understand a microprocessor? So this is a question that Jonas and Korting wrote about back in 2017, and they are also very much pushing right now the move towards using connectomics as a basis for the type of work that we're doing and something similar. Let's assume that you're using the same method. Here's the anatomy of the microprocessor. This is the 6502, very well-known processor. And here you've got, just as an example, just to compare, as a diagram of the primate visual system. There are many different diagrams for that, but this is one way of describing it. Let's assume that you have that. So you've got your anatomical description that identifies different regions. You have prior observed interactions, you make a hypothesis, and then you set up a study. Now your study is going to be something like, we want to record from certain components here. It's like, say, set of transistors. Have a behavior, for example, a program or a game. And then there's a manipulation. You might carry out a lesion study as you would do in neuroscience. But in this case, a lesion is you break transistor or turn it off, or maybe you give the transistor a voltage so that it's doing something it's not supposed to do. Before we dive into what the problems might be, what would be a satisfactory understanding of a microprocessor? This is related to what would be a satisfactory understanding of a brain. A satisfactory understanding means that you don't just understand the system, or at least to me, a satisfactory understanding would be not just trying to understand it at one level, not just having a description of different parts of the system and how they work with each other, but you want to have many levels of description. So here, for example, you've got this adder. It's one thing that you might find in there described at a lower level. Here you can describe it as logic, or you can get all the way down into the silicon. These things, these different levels, it might be useful for different reasons. So you might decide that you're going to work at one level, and that's where you're going to describe what's going on. You might use this one here, for instance, if you're thinking about programs. But what you don't want in your understanding is you don't want that your hypothesis, that your model, your theories about how this all works, that they work at this level, but then they break at the next. So the levels of description that you're using, what you're learning, your understanding has to carry through. It has to be something you can follow all the way through the levels. Okay, so here's what you might do if you try to get the connectome of this processor here. You put it under a microscope, you look at it, here you have an image. You can see all the parts that are there, draw them out. This is so similar to what you see in the connectome of the brain in a sense. I mean, you'll see it in a moment. There's so many similarities. Then you can take a closer look at some part here and say, oh, look at that. This looks like it's a transistor. So you've been able to pull something out of that processor connectome where you see something you know, something that you can identify and maybe even describe as in terms of equations what's going on there. All right, so what might go wrong? If you're a neuroscientist, one of the things you'll do is you'll have hypotheses about which cells are involved with what. So if I carry out a lesion, like I'll cut a piece of a connection, or I'll scoop out a bit of brain, what's going to happen to the behavior? And that tells you a lot. Well, here you could have some hypotheses about which program or which kind of programmatic behavior, like say, adding two numbers, different transistors on the chip are involved, got green, blue, and red involved with different things. At least that's what you're testing here. And then if you carry out those lesions, then you'll observe something. But if you understand microprocessors to some degree, you can already see where this is getting really difficult, where you could go very wrong because there are so many things upstream, downstream connected in some way. So what does it mean if you say, oh, this transistor lights up, or this transistor, you know, if I take it out, then suddenly it doesn't work anymore. It doesn't necessarily mean that transistor was carrying out your addition. It could just be that it's on the path leading to that addition in some way, right? So these are all typical problems. How about tuning studies, where you're thinking that you're a neuroscientist, tuning study means, okay, I think that when this cell fires in a certain pattern, or it fires at a certain rate, that means something. That rate of firing in that neuron means you're going to move your arm, something like that. Well, in transistors, you could do the same thing. You could look at high and low voltages, and you could have some idea that you're looking at a particular tuning there, that you see a certain sequence of high or low or pattern, that means something. But again, you can imagine how easy it is to be misled, thinking that looking at the tuning of that particular transistor really means something. So here, how about meaning? One of the things that we do in neuroscience is that we say things like a memory engram is a pattern of cells that fire together, an attractor. Neurons that all fire as a pattern. So this pattern means I saw a car, or this is a memory of a car, and here's a memory of my grandmother. But they're just neurons. It's a pattern of neurons. Why do they mean anything? You could just swap that and say, now this is a car, and this is my grandmother. Why is it in a particular way, not another way? And here, trying to do the same thing, you've got these games like Pitfall, Donkey Kong, Space Invaders. You're looking at patterns of activity on 3,510 transistors, and you're trying to discern meaning from that. Why are you seeing certain transistors light up and not others? This would be even worse if you're looking at RAM. where say you have an operating system that is constantly allocating programs in different parts of the RAM. So what does it mean if you're seeing it light up here one day, over there next day? It's going to be really confusing. And it's because patterns themselves don't have any meaning without context. Like those neurons firing for your grandmother, they don't mean that that's your grandmother. They only mean that's your grandmother if you know the decoder downstream that's using that pattern and the associations with other patterns. And then with things like saying something like, oh, hi, grandmother, those sorts of things. It's only in connection, only in the context of the whole that these parts have meaning. All right, so enabling causal neuroscience. You can already see that I was mentioning the whole there. What you really want is a full connectome. You want the whole, or at least enough of the whole to be available that you can trace what's going on all the way through. Because then you can understand meaning by looking at how things relate to other things. And you can also see what's firing first, what comes next, fourth. So what you want is a full connectome. You want a detailed circuit reconstruction, high level of detail, and you want to be able to trace things all the way from sensory input into mental or cognitive processing, how that's being influenced by memory or how things are being stored in memory, how this affects your experience, how your experience might be affecting what you're processing, intrinsic drives, and then behavioral output. So you've got this whole chain going on. All right, so here's an example of high throughput, large scale microscopy. This is data from the FLY Marginalia research. One of the big milestones that was recently achieved is that they finally published the whole fly brain. So that's the brain of one particular fly, not a whole bunch of different flies. One fly scanned completely. And what it means is not that they only just did that scan. Did that years ago. But right now they've gotten to the point where they've annotated it enough and done enough work on it that you can make it available to scientists. And you can see here that you can kind of see how you're going through different slices in the stack. You can look at it at different resolutions. So you can see either cell bodies, or you can see where the boundaries are between processes that if you look through the stacks, they create these dendrites or axons. If you zoom in really tight, then you can see the individual synapses, and you can see the little vesicles that contain neurotransmitter. And if you can count all those vesicles, you can get some notion of what the relative strength of that synapse is. So this is the sort of data, raw data that comes out of that. And then you can do something like this. So alignment, segmentation, identification, 3D reconstruction. So you were already looking at these images just there and how you're making your way through them. And then as you can see, if you're doing good segmentation and they all line up nicely, and you can distinguish one thing from another, then you can find axons, dendrites, and blood vesicles. So you can find all sorts of structure in there. But the important thing to mention is that this is still just a static 3D structure. So it's beautiful. You can see so many different components in here. And you can pull out different parts. It's going to zoom into that in just a moment. But it's still just a static representation. Now we have this from the whole fruit fly. We also have a bunch of data from the mouse brain, not a complete mouse brain, but there are now 6 separate 1 cubic millimeter chunks. They're from different mice that have been done at the same resolution as the fruit fly here, although the quality isn't quite as good. There's not as much contrast. The fruit fly data is still better. There's also been 1 cubic millimeter of human brain that recently came out of Max Planck Institute that was in the press everywhere because it was human, right? But it's basically the same sort of data. And then now there's a whole mouse brain connectome project where it's going to be not just a cubic millimeter, but the whole mouse, also electron microscopy. Yes. And in the 1 cubic millimeter of mouse brain, I believe it was 2.5 million or something like that. I'm not 100% sure. I could be wrong. Yeah, it has 140,000. That's right. Yeah. I believe so. Yeah. Yeah. Yeah.
I'm sorry to interrupt. Can you please repeat the question? We cannot hear it on Zoom.
The question asked was how many neurons were in the cubic mouse millimeter.
Cool, thanks.
Sorry, cubic millimeter mouse. It's not like a cubic mouse. That would be a very interesting mouse. All right, so just summarizing a little bit, to get to a proof of concept, you've got your data. You need to create your static connectome data there, and you identify cell types. Then there's the translation to a working dynamic model, and then you need to do validation. All right, let's see some examples of attempts to functionalize. This is a very brand new piece of science, a very new area. There isn't that much. There was some prior work in trying to do reconstructions of just single cells and to have a functionalized version of them that was a good representation of how, for example, pyramidal cell fires, et cetera. But there are only a few studies from maybe a handful of labs right now working on functionalizing the kind of data that we just saw. And to functionalize, what you can do is you can look at, obviously, the shape of what you've got. That is something very useful to identify, is this a pyramidal neuron? Is it an interneuron of some kind? What type of neuron you're looking at? But you can also use information like location. Where in the brain did you find this? What is it likely to be? So if you have multiple things that have a similar shape, well, where did I find it? What could it possibly be? Same sort of thing with synapses. If you've already identified two types of neurons and they have a connection, then you know that there are probably only n different kinds of synapses that they can have and different types of ion channels in those synapses. So location tells you a lot. Once you have an idea of what you've found, you've identified your cell, then you can use libraries that have been created that have looked at in vivo, that have done recordings in these cells to see how do they respond, describe the response, and then you have a probability distribution of those responses. You can use that as part of your information to functionalize. You can do direct measurements. So for example, I was talking earlier about using compartmental models and then directly measuring, say, the thickness of a dendrite and using that to inform what you're putting in. You can count vesicles. And then you can use constraints. So constraints that are based on the neural code to simplify your problem, to simplify your functionalization problem. So neural code can be, for example, is this about a cell that is somehow very important? It's a single cell that is sending information everywhere, that is firing with bursts, or is it a population code that you have? Are there many cells working together? Is there typically a brain rhythm present so that firing can only happen at certain phases of that rhythm? Is there recurrent inhibition that stops all firing very often so that you don't have to care very much about the details of the response function right outside of, the beginning of the firing because it's being inhibited? So there are all sorts of things that you can use to try to constrain your problem of functionalizing further. So our first example is Philip Xu et al.'s paper that just recently came out in Nature, where they functionalized a fly connectome in a very simple way. They took the connectome, they knew which connections were likely to be inhibitory and which were excitatory. They treated them very simply. For instance, here they just used a 1 or a -1 as excitatory inhibitory, multiply that with a weight. The weights were also obtained in a very simplistic manner. What they did is really just use the same model, every neuron, in a leaky integrate and fire circuit. So very simple spiking neural network. And with just that very simple network, a simple network where they also said everything returns to the resting potential. So everything is like null normally. If we activate some cells up here, what can we say downstream about the neurons that are going to be involved with that? And if we look at what we're saying here, can we check that? Can we match that to something that's been found before? So can we say that just this very simple way of looking at the connectome and functionalizing it, can that give us the ability, say, to make predictions about neurons in a similar way to the way the older correlational studies were done, where you would look in vivo and probe at a neuron? Can we predict that? And so what they did, that exactly, and they ended up with this beautiful schematic. It's much more detailed in the paper, but what you end up with is you can follow from input to output what's going on, at least, within all the limitations of that super simple model they made. So it's the same thing I was talking about before, from sensory to motor to output to behavior, following it through and through the intermediary stages as well. And they were able to predict that this MN9 neuron fires to elicit the extension of the proboscis so that that fly can try to get some sugar water. And that is something that matches the literature because that had previously been found. They were also able to show that there's another cell that inhibits that, but only when there's a weak stimulus for sugar water. And that again matches what they previously found. They were able to show that there were two different pathways, but they shared some components for sugar and water seeking. They operate additively, and they were able to identify neurons involved with the grooming of the antennae of those fruit fly. If you want to see all the details, please go see their paper. What it showed was not anything new. They didn't really make any discoveries here. They just showed that even with a very simple model, it was kind of surprising that without getting into more detail, you could make some predictions that were at least interesting enough that you could say, wow, this is something that if we had had that years ago, would have really helped in those studies to try to identify what those cells were about. And you have a causal explanation for how this pathway works. Another study that we recently did a journal club on at Carbon Copies is quite different. They did something they called connectome constrained networks, meaning they used the connectome as a constraint on a network that they built to try to predict something about the visual system of the fruit fly. So here's their claims. Like if they train this connectome constrained model, they do the training for a specific task, that it helps you identify circuits involved in the task. That you can then also validate that by looking at 26 other studies that have been done earlier. This is the schematic of basically what they're doing here. You've got visual input fruit fly. The fruit fly then has neural activity in certain parts of its optic lobe. You have the model that they build, which I'll say a little bit more about in a moment. And again, you see activity in some of the cells in that model, and then do they match? Neurons that are this particular neuron in the model, are they doing what you see in the fly, that particular neuron? And there's a lot known about those neurons. You can identify them by name, helps this process here. This is a very busy slide. I won't get into all of it, but what you have here is you've got their connectome. This connectome, as you can see, you've got all the different cells on the side here and again here, and it tells you what's connected with what. The size of those blotches indicates the strength, and that's a synapse count that gave them the relative strength of the connections. You can see that they are extremely schematic here, and that's because what they did is they did not use the connectome in its unique entirety. They didn't try to parameterize everything. What they did is they looked at the circuit that they knew about. That's this circuit here that where input from the retina is going into these lamina and medulla areas, and then from there is being processed in the lobula and lobule plate. They use this as a very schematic thing where they're saying, okay, we're going to assume that this visual system does a convolution. We've got the same lattices over and over again, and you've got connections from one layer to the next going through like a column and then spreading out to nearest neighbors to some degree. So what they did is they took one part of the circuit that they knew about, and then they averaged. They looked at that same circuit in different parts of the fly brain and averaged over that to see, okay, this is what the connections usually are, how they spread out. Here's how many synapses you normally see. They use these averages here to make what you might call a stereotypical circuit. And then they replicated that stereotypical circuit many times. So this is a synthetic model, but it's constrained by the connectome of the fruit fly. And then they added to that the task training. So they trained it to do things. You have here examples of these cells like the neuron R1, neuron C3, neuron T4B. These are types of neurons, so types of cells. And the same cell, the same type of cell occurs many times in a lattice. You can see that the lattice spacing here is a bit different than in this cell type. So R6 is a cell type, not a single cell. And then you have visual input coming in, and then you can identify that some of the cells are responding to contrast changes and some of them to motion, some of them to both of them combined. And that's where they make predictions then. This making predictions based on this connectome constraint network where the free parameters were trained to the task. And what they were really interested in is getting those free parameters down because the functionalization problem is so big. If they had a completely unconstrained network, they'd have over 2 billion parameters that they needed. But because they use the stereotype system, the stereotype networks where everything is over and over again, it's the same, they only had 734 free parameters, made it way more tractable. Not going to talk much about this slide. Let's just skip that one. Sorry, yes, go ahead.
Are those weights in these networks, are they informed from the connectome? Like it's basically that you showed earlier here,
Yes.
showing how many synapses are connecting each cell type to each other cell type.
Right. So they looked at many examples of the same circuit because they were, over and over again in the fruit fly, they saw the same circuit, and they would measure how many synapses do I see here between this neuron and that neuron? How many do I see here between the same type of neuron and same type of neuron? And then it would average. So the average number of synapses seen between neurons, that is what gives you the weight then. So you have here these synapse counts that are represented here in the size of these connectomes. So yes, that's what they did. Okay. So let's summarize the results of that. One thing that was really important is that they figured out one way to reduce the number of free parameters you have to try to estimate because that's so difficult. The simplifications they used here probably work in a number of different places. And one example is the fruit fly visual system because it's very stereotyped already. And this is great if you're going to do very testable causal hypotheses and test those. But you can already imagine that there are some limitations here. So one thing that they found was very important. They did some comparisons on artificial networks where they tried to use the same method. That sparsity is very important. A sparse system is much easier to work with for this. It's very important that you indeed have convolutional homogeneity, that you can have these stereotype subcircuits. And if you have a region of the brain where there is more training, more learning, more specialization going on, then you might not have that. And in the human visual system, for example, you might find that there are already a lot of specializations that would make that much more difficult. Yeah, and okay, there's the issue that what they did, of course, is they trained it for one task. So this does not generalize across tasks. And if you do try to generalize what they did here across many tests, then you might find that you need many more parameters. So then you need more details from the connectome to be able to represent those parameters. Okay, if you want to see a lot more about this, again, please go to our website, especially the braingenics.org website, where we really dive into functionalization. Okay, I wanted to say a few things now about synthetic data and why synthetic data is useful in this case. The really important thing as we're trying to develop methods for functionalization is to be able to validate those methods, to be able to discover quickly what works and what doesn't. You might compare that, say, in machine learning with testing an algorithm and seeing how well does it really perform. How does it compare with a different method? Which ones are they strong or weak at? What are their strengths? And why would you choose this one over that one? What could you tweak here? Those are the same sorts of things you want to do in neuroscience in trying to functionalize these piles of data. But the problem is that for good functionalization, you need ground truths. It's like in say, computer vision, you have previously annotated data sets. You know that you see a bicycle, you see a tree, you see a car, even if it's obfuscated, or you can have them at many different levels of difficulty, but it's an annotated data set. You have a ground truth. It's something you can use for validation. In neuroscience, the typical ground truth, if you go to a neuroscientist and say, oh, I want to make synthetic data as a ground truth, they'll laugh at you, typically, because ground truth is biology. You can't have a synthetic ground truth. That's ridiculous, right? But the problem with the biological ground truth is that it's not a fully known ground truth. You don't understand it. Sure, it's there. There's a piece of biology. You don't know what's in it. You don't know what it's doing. You don't know what the circuit means, or when you've correctly copied its function, when it's doing the same thing as that circuit's supposed to do, missed something important. You just don't know. Now, C. elegans, that little worm, might be a possible fully knowable system because it's so small. That's the reason why some labs, like Conrad Cording's lab, the same scientist who did the whole thing about microprocessors and neuroscientists, they're focusing on C. elegans because they want to dig down, they want to get so much data about that biology all the way down to the molecular level that they can put it all back together again. And because the worm has so few behaviors, that they can correlate everything with behavior. They can see what the worm is doing, record from neurons using calcium imaging, voltage imaging while they're alive. So they're looking to make this a knowable ground truth. But in the meantime, synthetic data is a good way to go. And the reason I put that here is because in AI, for example, it's quite normal that you often use synthetic data sets. Like, before you send a car out on the road to drive by itself, you probably want to try that on some synthetic data first to see how well it's doing. We don't have to get into this. You guys know about data sets. All right, so here's something related to our project. This is a project that has recently been started at the Max Planck by Jürgen Kornfeld and others. It is an attempt to make synthetic data, so synthetic electron microscopy data stacks. And the point of this, this platform, their version of it, is that they want to use those synthetic data stacks to improve methods of segmentation. And they can do that because you know what you put in. You know that you put cells, neurons, dendrites, axons, all sorts of things are in there, glial cells, vasculature. And then when you're segmenting, you can tell whether you did it right and where you're going wrong. It just helps. So this is exactly what they're doing. The difference between what they're doing and what we're doing in our platform is that they're interested in segmentation and we are interested in the functionalization. So we're interested in that extraction of function, the emulation. So our brain emulation challenge platform, this is the last part of this talk. It's an open source platform that is meant as an initial path dealing with the missing fully known ground truth problem. To use that to validate both structure and function emulation. So two different things that we're trying to validate there. And we invite everybody, researchers at university or anywhere, to participate. So we want everyone to use our platform as a way to help develop their methods, but also then, of course, to critique our platform, to improve the platform and support. Everything is a one big iterative cycle. We aim to have competitions, and everyone gets back a detailed error report telling them exactly where their attempt at emulation went wrong, so that it helps improve what we're doing. So the improvement loop we're trying to get here is this. You begin with collected data. You end up making your static reconstruction. There's been so much work on this. It's not perfect yet. There's a lot of human proofreading that happens to identify cells and ion channels and synapses and whatnot. That is still cumbersome, but it's, it's really good. Then there's that translation step, build a dynamic model. And then what we want is to be able to give you really good verification and validation. So this is really what it's all about. Getting that back, prove your methods, and so forth, and so forth. There's an equivalent to this improvement loop. If you look at, say, machine learning or AI or computer vision, they all have loops like this. And our brain emulation challenge is like this. So you start off with some intended function. We like to aim for systems that carry out a task that is related to something that is cognitively relevant, because then we know we're talking about a level that we most care about. Because so, for instance, I don't know which synapse is firing right now. I don't know which ion channels are firing. Personally, I don't care too much about that. I care about the cognitive function. I care about being able to remember things, being able to retrieve memories, being able to recognize people, have certain interests, all that sort of thing. So cognitive functions are really the key. So we want systems that have some kind of cognitive function. Maybe they can remember something or do something else. We take that and we describe it as cells in enough detail so that we can then extract an artificial data set that looks like electron microscopy, another one for recorded activity in cells, and we also do calcium imaging. So we provide these data stacks. Now, this is what the brain emulation challenge makes available as the data for the competition, data that anyone can get. They get that data stack. They do not get to see this, the ground truth system. There are example systems they might see, but they don't get to see this for the challenge. Then the challenge taker takes the data stack, carries out the 3D reconstruction, translation. Here's my emulated system. This is what I think I found. They give that back to us, and we can validate by comparing directly with the ground truth. So for example, for structure, you might find that, oh, look here, there's a connection missing right there. No, and here, oh, look, there's a connection that's missing in here. So there's some errors in this thing. The same way we can also compare function, because if you have a system that does something specific, like say it stores a memory pattern, and you can retrieve that memory pattern, then we have activity we can give to the system that does that. That's what we put in here. And we can use that same activity to test what's happening in the emulation. We can also do sensitivity analysis and a variety of other functional tests on the system. So I already said most of these things, so I don't have to get into too much detail. All I'm going to say is that as we pick our functions, we can go in many different directions. So we can do something auto-associative, or here, super simple. This is not something that you would think to find in the brain, but it's a really nice example for presentation because it's so simple that you can understand it when you look at it. Spiking XOR operation. Okay, so first we grow realistic connectome reservoirs. And actually the next slide is better than this one. This one shows you two different regions and cells that are being developed. You've got axon outgrowth. This is all based on a platform called NetMorph that was published back in 2009. And that platform describes how growth cones on axons and dendrites work. And so that you can develop cells. And by tweaking parameters, all sorts of parameters about elongation, branching, and so forth, you can develop neurons that look like pyramidal cells, or stellate cells, or chandelier cells, all kinds of other types of neurons. And in any case, if we put them all together into populations here, we can make layers. We can have those layers connect with one another, find places where the synapses might be. This is just another quick video of outgrowth of cells between 3 different layers. You end up with, oops, that wasn't meant to happen yet. Back here. This is the one. On. Okay, so this for the XOR, this is the input layer here. Then you've got 2 layers in the middle. You've got one that is excitatory and one that's inhibitory. And then you've got an output layer. I could describe how using reservoirs of excitatory, inhibitory neurons, and then having those layers there, how that can give you a spiking XOR. We get into that detail, but I think if you want to, you can even see it next. So after building the reservoir, you can prune and tune the connections to get the circuit to do what you want. Now, you can see that, I don't know if you can actually see it, but let's see, you've got the input here. You've got a spike on A. Then you've got, let's see what happens. The output. Oh, here it is. So if you got one spike on A, but you have no spikes on B, then you've got a spike on the output. You've got one on B. You've only got them on B, not on A, same result. You've got them on B cells and on the A cell as well. You don't have one, so it's doing an XOR here, 1, 1, 0. And you could look at these layers in between to see what's going on. But in any case, this is just, like God's eye view. We have access to the whole thing. We can just analyze it. It's, it's fun. And again, I hate it that this happens. Why does it do that? Right. I just want to see this bit. Okay, so we're almost at the end. After making the connectome work in the reservoirs that you've built, then you want to make something that is a complete set of virtual tissues. So you're not going to have only the cells that are part of your function. There's going to be a lot of other stuff there. There are additional neurons, there are glial cells, there's vasculature. There are many, many things that you can stuff in there. Nice thing, of course, about a system like this is you can make it as complex or as simple as you want. So you can either not add anything extra and make it very easy to find the cells that are involved with the function, or you can make it harder. And then you make these stacks of data, right? Your EM, your calcium imaging, your electron microscopy. And then you can do something like visualize your electron microscopy stack. This still needs a bit of beautification in here because there are many breaks in the dendrites. And then here you end up with a challenge output. This one here is fictional, obviously, because it's from 2025 from the company We Reconstruct LLC. But yeah, so we gave them back information about their structure validation, and then down below about their functional validation. We do things like giving them the graph edit score. Graph edit is when you look at, okay, to get from what you give me and what the actual structure is, where do you need to remove a connection or add a connection? Where do you need to change the type of a connection? Where you need to add a neuron, remove a neuron? You can develop a score based on that. It's one of many, many graph scores. So summarizing, yeah, with the ground truth fully known and can focus on the performance for a certain cognitive function. So we're focusing at a certain level. You know, we talked about scale separation at some point, at a level that we're most interested in. We can use competitive scoring and precise feedback to help improve how methods are developed. So that's the closing that improvement loop. We have full control because this is all, you know, we're making this thing. So we can make it as simple, as easy, as difficult as we want. We can look for different types of networks that are where maybe the challenge is something totally different because the cells are doing something very different. Coding, the neural code being used is different. We can extend beyond EM. So for instance, expansion microscopy is getting really popular right now. If we want to make data for that, we can do that as well, and so forth and so forth. And really interesting is that you can explore the problem of scale separation explicitly because we can decide what resolution we use to build these models. So we can see, does the model resolution really matter in terms of getting back what you put in? Or if we make the model that we put in have a certain resolution, what can you abstract out of it, or what do you still have that you need to look for? And the same with the data. So the data resolution, if we make the data resolution better, worse, or we add noise, what does this do to the problem? So there are many things you can explore, you know, with a real example right there. The main risks of something like this are, of course, number one, if you have a competition, so you have a set of criteria scoring for, people will always aim for those scores. So you're going to optimize towards a score. If you want to make it really aim at the goal that you have, then you need to have enough criteria so that you cannot really win unless you're actually aiming in the right direction. There's no real way to get around that. This happens in any game, any competition, anywhere. But if you don't have a score, well, you're not really doing something quantitative. So what can you do? This is just a risk. The other thing is built-in assumptions that we have about neural coding. Because we make a system, we build a circuit, we have some assumption about how those cells are connecting and communicating with one another and what matters and whether they have brain rhythms that are modulating their activity and things like that. So we're building in assumptions. And so the problem is Randal neural code really anything like real neural code? Where are we going wrong here? We're building into this competition, into the challenge, something that people have to then find that may diverge from reality. So the only way to get around that, because of course we're doing our best, just like people who are collecting the data are doing their best to collect the data that really matters. People who are trying to emulate, who are trying to functionalize, are doing their best, focus on the things that really matter, but they also don't really know how everything in the brain works. And the only way to get around that is to do all of this iteratively, to keep on working on the same problem, either as building the challenge or doing the emulations, and then deciding, well, here's where we just discovered that things don't really work that way. You're going to have to adjust something. Again, there's no real way to get out of that problem, how to get around it, but that is something you have to be aware of. That was it. And also, I just want to invite anybody who's interested in what we're doing, please do reach out. We are always looking for more people to join the team. Thank you.
Thank you so much, Randal, for the talk. Do we have any questions? Anyone? Questions?
Great talk. I really enjoyed it, but I had some questions about what do you think the importance of cell types can be for computation in the brain? And also, how would you define, or what would be the data modality that you think it's most important to describe cell type? Because as you said, you can go for location in the brain. There's single cell transcriptomics. You can also go for electrophysiology measurements to define the cell type. So I was Just wondering if you think that there's a more important cell type classification for computations that can be, yeah.
So cell types, even just classifying cell types in a variety of different ways. And so the original way that cell types were classified, say when you go back to early studies, we're looking at cells that count, is largely on the appearance of a cell. So it's something where you can say, oh, this looks like a chandelier, so this is a chandelier cell. Looks like a pyramid, it's a pyramidal cell. So you've got your cell types there. To me, what matters about a cell type is what is the pattern of the connectivity that it makes? Which layers does it connect with? Which other types of cells? So where does it fit into a circuit? But also the synapses that it makes, what kinds of ion channels do they have? And why do I care about those? I care about those because ion channels have response functions of some sort. And the response function they have determines their temporal response. It determines whether they can fire and then have to wait a long time, return back to base levels, or whether they can fire very rapidly. It matters whether they're inhibitory and excitatory, of course. There are many things that are about the dynamics or about the architecture that I find are most important when you look at cell types. So if you're asking, do cell types matter? To me, that's the thing that matters because I care about function. I'm sure there are other things that one could care. So for example, if you're interested in disease, then, certain cells will be more likely to, for instance, die off in Alzheimer's or in Parkinson's than others. And that's going to matter. But this comes back to this whole question about emulation. You do it for a certain purpose. So I care about this aspect of cells, not about that aspect. I don't care about the patchwork that nature used to build the cells and what it's then subject to in terms of disease or not. Not necessarily. So that was part of your question is, what does it matter cell types and what do you care about in terms of cell types? What was the other part of the question?
I think it was all related to that. Like, what do you think the modality that's the most important to get this information about the cell that's related to computation is? So in your answer, you already said that you think more that the electrophysiological features that depend on the ion channels are the most relevant. Yeah. Thank you.
Good questions.
Hi. Yeah, I appreciate the talk. It was really interesting to hear about the whole brain from an emulation kind of standpoint and all the connectivity and all. So I have a question kind of back about the actual the causal neuroscience and how this informs the causal neuroscience compared to like kind of an associative approach. So like how does this kind of close the loop and make it more causally informed, I guess?
Okay, so I think that to be fair, there is never one study or one experiment or one way of doing things that is fully correlational or fully causal. You're always going to make some causal description, test some causality in some sense, and do some correlational work. In fact, you could say that at some level, when you get down to the description of the components you're using in these networks, in these circuit networks that you're building, that once you get down to the lowest level, you're always working on basically a black box where you're just correlating what's happening input output. So that always happens at some level. I think the distinction that I'm making here between causal and correlational neuroscience is a neuroscience that largely depends on making statements and publications that say things like, we have found that this area is involved in, we have found that this neuron is involved with, that sort of statement, right? Rather than saying, here's our description of a mechanism that shows how this happens. Now, that's why I also said there's never usually, hardly ever, something that's completely just correlation or causation, because often there will be a description of some sort of mechanism or at least one mechanism, but not a full description of how something works. So the biggest change is a desire to have a very causal description. So a description with mechanisms at a scale where you can begin to follow the data that comes in to the behavior that goes out, and you're trying to describe exactly what's happening there. And again, this is a vague statement. Exactly could be determined at many different levels, right? I'm not sure if I'm explaining myself.
Yeah,
okay.
yeah.
Does that give you an
So
answer?
it
Yeah.
seems like also from having a kind of model fit and applied in some way with maybe system identification or something, you get to be able to see how mechanisms can happen that give you similar kind of answers as you would have gotten with that kind of experimental approach originally. So it kind of ties it all around.
You could definitely close that loop. You can get the same. That's in fact what they're doing in these experiments, right? When you're seeing the study where they did the constrained, connectome constrained network, for example, and also Philip Hughes network work, they ultimately then validated what they did and showed the value of their mechanistic explanation by comparing back to those correlational studies that were saying, oh, this neuron here seems to be involved in detecting changes in the lightness or darkness of whatever is being presented to the fruit fly, or movement or something like that. This cell detects movement. And they were able to show, oh, yeah, okay, our circuit is showing how this cell detecting movement. But then there's that little bit, right? The how. It's not just it detects movement, but it detects movement because these other cells that we're detecting, either gradients going that way and gradients going that way, we're combining here, and that got put together. So you get a description and explanation of what's going on beyond the correlational study. But it definitely, they use those studies then to validate their method. And then in the next step, you take the same method they used and you make a prediction for cells that you have not yet studied in vivo to see how they respond. So then you're trying to inform experimental studies where you go back to the living fruit fly, you record from a cell and say, hey, so was that true? Should we look over here? Should we look over there? Model says we should look over here. And then you can hopefully find what you're looking for.
Perfect. Yeah, I appreciate it. Thank you.
Any other questions? I have a question actually. So imagine if tomorrow we have the ability to emulate an entire human brain, maybe on a supercomputer, maybe with like crazy parallelism and so on and so forth. What could that really give us? What can we do with that ability?
What could you not do with that ability? So yeah. Well, the first thing, of course, that we were just talking about is understanding, right? A mechanistic, detailed causal understanding, so you can understand, okay, what is going on when you're thinking? What is going on when you're remembering? What is going on when you're feeling? What is going on when you're empathizing, when you're modeling someone else? These sorts of things, small things, big things, start to unravel that. So it's for learning, for science, obviously. Another thing you could do is you could learn about ways in which the brain is combining many different algorithms. So for example, memory in the brain is a process that goes through many stages and filters. And there are different types of memory. This is much more complicated than the sort of thing you typically see memory in AI. So why is that? What does that give you? You might learn something about algorithms that you can use in AI by looking at the brain. You can also build devices. Say, for instance, if you have patients where they have a damaged hippocampus and they need a neuroprosthetic that can help them replace the function of the hippocampus that is essential to being able to form new episodic memories. That's the sort of thing you could help build using something like this reverse engineering. And in principle, you can go beyond that and say, well, at some point, if you can emulate a brain and you have a brain in a computer, that is another way of, say, existing. Say it's a new embodiment of that mind, right? So then you have a mind in a robot, or a mind in a spaceship, something. So it doesn't have to be a biologist. You could take that all the way out to that future. You can imagine what does that have as all sorts of ancillary consequences.
Fascinating. Thank you so much.
Thank you for your talk. Those are really valuable insights. So I wanted to understand how these like really micro scale molecular modeling scales up to a bigger system like the brain, and how this can inform computation through dynamics and more of a dynamical approach to understanding the brain.
That's a very open question. Can you specify what you're trying to have me explain to you there, or to?
How does your model scale from the molecular level into a whole brain level?
So that's a question about how you scale up, is that really? Or
Yes, and does it really scale up? Does having these super fine-grained gradients for individual neurons and individual gates really scale up to whole brain resolution?
Okay, right. So yeah, this is actually super interesting and is directly related to some of the things that I was pointing at beginning, which is the question of scale separation, but also the resolution you're supposed to take into account and emergent phenomena. Because the idea behind something like brain emulation is, in fact, that if you go to a fairly high resolution and you replicate all sorts of tiny components and you put them all together, just as you would put transistors together into a chip, you then get the behavior of the chip, same way you get the behavior of the whole brain. Would it work? We assume it would. Why would it not work? Why would you not get certain types of emergent behavior from it? Well, if there's something missing that you should have been including, that you did not include because you thought it wasn't important. So for instance, let's say that you have memory at a synapse. That memory is composed of different stages such as LTP, and LTP is a molecular process. And then there's some aspect of that molecular process you didn't include because you decided that only connections and spikes are important. Then maybe you were looking at the wrong level. And that's the discovery of the level at which you have to do that scale separation for a particular part of the brain, for a specific function, some aspect, some feature of what it does. And that's not something where the answer is no. That is something that is still an exploration. So that's why these types of platforms are meant to help explore where you would put that level of scale separation. But I should say so. That's why I started off saying this is all based on a philosophy of functionalism, because the underlying idea is that, yes, you can do this reductionist approach where you, if you just look at the right pieces, if you include the right bits of information, it's like if you're trying to do reverse engineering of a computer chip. If you look at the right pieces and you put them together, you will get the combined behavior. You will get the emergent. There are all kinds of reasons why that might not work. And a really fundamental one would be if the functionalist philosophy behind it is just completely wrong, right? So it's worked so far. It's been something very helpful in science, but you know, might not be wrong.
Okay, appreciate it. Thank you.
Any other questions? Okay, if you want to talk to Randal more, we have, I think he will be around for how long, or a few more hours? And
yeah, I wasn't told how long or anything. I just said I'd be available. So
we have a few slots open, I think still. You can email. And oh yeah, and I wanted to say also, if any of you are interested to get involved more with the project, also you can also contact Thomas here. So he can he's planning to start a student sort of group, like a research group, and then they can work on this and more. So get in touch with both Randal and Thomas. Can we have one more question?
Yeah, question.
Yeah.
So thank you for the talk. I don't know much about neuroscience, but from what you showed the static images of the brains that you had for fruit flies and of the mouses as well, or the mice. And it's my understanding that brains, like neurons, typically can be dynamic, like rewire and change shape. And so how does that kind of complicate things versus like a computer chip, which is like static, right?
Yeah, very much so. So of the many parameters that you have to functionalize, or you have to find the right values, they're not just parameters about the response function. It's also parameters such as in your learning, you know, if you have some description of LTP, long-term potentiation or long-term depression, and then how that transitions into longer-term storage, the growth of new synapses, the growth of new little neurites that connect. Those, of course, are described by equations as well, which gives you extra parameters that you then have to fill in. And getting these dynamics over time, long-term dynamics that change the structure of the system, they're even more difficult to find, put in. That's why most of the things that we've talked about right now are about a system that is not learning, that is not evolving, but that is static, just because no one's gotten to the next level, let's just say. But yes, that is absolutely necessary. I thought, this is another thing, I thought that you might have asked, because you mentioned, oh, you've shown all these static images and going from there to functionalization, you might have asked something like, well, why did you pick that? Why didn't you talk about, say, looking at function, looking at, say, recordings in cells and how you go from there to description of the system. I know that wasn't your question, but I'm going to mention it anyway because I think it should have been in the talk. So the main reason is because it's technically so difficult. The resolution we're interested in, and also the scale. Now, if you wanted to get recordings from all of the neurons in an organism, and you wanted to be able to observe them at a temporal scale where you're catching every spike, and you want to observe them over a long period of time, there are only a few organisms where you can do that, like C. elegans, or maybe the zebrafish. In the human brain and in chimpanzees and so forth, that is just not doable right now because the only way you can get the resolution temporally, also spatially, that you want to look at is to put electrodes in. And large batches of electrodes, first of all, they disrupt the brain. It's a lot of neurosurgery. Nobody likes neurosurgery. And then you're recording. And what you can infer about the system, about what's inside, what's going on, depends entirely on what you've recorded. So what was the stimulation given? What could you therefore observe and then infer? Have you observed long enough? And what if even observing what's going on in an organism over its entire lifespan isn't enough? Because there are things in that brain that were put there by the blueprint of the brain that never ended up causing something you happen to observe, but it could have in the future. So the problem with this sort of black box system identification, or gray box if you can manipulate it, is that you're only seeing a subset of all possible input-output behaviors, and then you're inferring from that. Whereas if you actually look inside the box, the architecture, the schematic, it's like with your CPU, that gives you a lot of information about what could happen, about latent possibilities. It's the main thing. Sorry to
Thank you.
take that on. It's just interesting.
Right. Thank you so much. That's the end of it. Yeah, I will. Yeah, thank you. Thank you so much.
Really enjoyed it. I should not run away with this.
Yeah.